My name is Susan Law. I'm the Knowledge Mobilization Lead for Long COVID Web, and delighted today to have Dr. Angela Chung here as well, who's the PI for our initiative. And we have two wonderful speakers for you today. So we'll get started with those in just a sec. We have some introductory slides here, and then we will turn it over to the speakers. I will start with a land acknowledgement. We wish to acknowledge this land on which the Long COVID Web operates. For thousands of years, it has been the traditional land on the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. Thank you. Just a recap of the purpose of these webinars. First, to share evidence-based information, research, results relevant for decision makers, researchers, clinicians, patients and families, and the general public. Second, to provide ongoing updates, spotlights on emerging evidence and scientific information about the activities of our national network. And finally, the purpose here is to contribute to reducing and managing misinformation about long COVID. We do have a code of conduct that's on our website. The link is provided in this slide. And at the core of it, it states that we value highly engagement around the science and its interpretation as well as its application. A commitment to reasoned discourse, as stated in our code of conduct, comes with the responsibility for all of us to ensure that any comments or questions remain in the spirit of positive, constructive, and respectful discussion. And we do welcome any and all questions and comments in this spirit. Logistics, we have a new platform. We're on webinars or Zoom webinar, and this provides us with extra security for these sessions. It does require that you log in and will be taken through a registration through your Zoom account. So apologies if this is a bit of an inconvenience, but I assure you we feel that this is very important to ensure these sessions are conducted appropriately and securely. We do have changed one of the logistics pieces where in the past you were able to post questions in the chat, and we still have a chat, so you're most welcome to put things, comments, reflections, questions for other people in the virtual audience. But I will be taking questions from the Q&A function, which is at the bottom of your screen. So we will pause for Q&A. I will read the questions, and the speakers have a chance to respond. These sessions are recorded, and we do have permission from the speakers to post these online following the webinar. And we do, of course, welcome your thoughts about how these sessions could be most helpful to you. We have a very short survey, which we would welcome you to complete at the end. So thank you very much. I will turn it now to the speakers. We are going to start with Professor Dr. Birgit Sawicki, who is a university professor and chair of translational immunology at the Berlin Institute of Health and Charité University of Medicine. She is going to talk to us about the immune endotypes and mechanisms driving the post-COVID syndrome. So over to you, Birgit, and thank you so much for joining us today.
Thank you very much for the nice introduction again. I will share my screen. Yes, should work now. As Susan said, I would like to talk about immune endotypes and mechanisms driving post-COVID syndrome. I did a lot of research in the early phase of the pandemic on acute COVID and the differences or the understanding of the mechanisms driving severe forms and the difference between the immune response in children and adults. But then was asked by Carmen Scheidenbogen, who is the lead here in Germany in actually post-acute infection syndrome research, to also dissect or help her in dissecting immune mechanisms driving the development or maintenance of post-COVID, and particular the most severe form, which is ME/CFS, and here defined by the Canadian consensus criteria, as you might know much better than I do, as I'm not a clinician, where you need to where the patients are suffering from fatigue, pain, post-exertional malaise, sleep dysfunction, and any of the additional symptoms here listed in these circles. But even in ME/CFS, we believe that these symptoms are not or develop on a different heterogeneous background of pathomechanisms, and so far, and this is what has made it very difficult to develop therapies, better diagnostic criteria to identify patient subgroups. And this is what we investigate here in Berlin, but also in a Germany-wide consortium. And the mechanisms proposed not only underlying ME/CFS, but other severe forms or long-lasting chronic forms of post-COVID syndrome, and potentially also other post-acute infection syndromes, are manifold. 5 just listed here. Persistence of viral infection, dysbiosis of the microbiota, endothelial abnormalities, but also dysfunctional neurological symptoms. And in the middle, we believe that at least in 1/3 of the patients suffering from ME/CFS, there's an autoimmune process ongoing. But if these mechanisms are listed here in separate boxes, this is really not true and a little bit unfair, because of course you have overlapping mechanisms that viral persistence can cause autoimmune processes. And of course, autoantibodies can contribute to dysregulations in neurological signaling if the autoantigen is maybe an antigen expressed by neurons. And to understand that and what drives then the development of autoimmunity, I've put here up a small cartoon where on the upper row you see the process how a controlled immune response would lead to clearance of the virus or bacteria. But then you get a decline of the immune response, dampening, and normal tissue regeneration processes step in. But in some instances, this might fail, that you don't get a complete dampening of the immune response with only immune memory being left, but an active inflammatory process continuing. It doesn't have to be a really drastic, severe one, but it could be a low inflammation process, which leads to a chronic inflammatory response, and could drive autoimmune processes, where what I try to to visualize here with the cartoon, where you need an interaction between certain immune cells, that's T cells, helping the B cells to differentiate into antibody producing plasma cells. And in the case of an autoimmune response, the antibodies produced by these plasma cells would target autoantigens from endothelial cells or brain antigens. So just depicted. But what's the evidence? Carmen has actually done some long work on trying to identify lead autoantibodies, targeting lead autoantigens in ME/CFS, and in particular here post-COVID induced ME/CFS. And you can see that, for instance, certain antibodies binding to G-coupled protein receptors like the beta-2 adrenergic receptor here are stronger correlating with ME/CFS than non-ME/CFS driven post-COVID syndrome. But what this cartoon or this figure already shows is that you can have many different autoantibodies, and this is what made the association with autoantibodies really driving ME/CFS or post-COVID syndrome very difficult. And I come back to that in a minute. What's other evidence that there are autoimmune responses driving the disease? They come from interventional trials where clinicians try to target either the B cells, which would develop into plasma cells, or the plasma cells themselves by giving, for instance, daratumumab, an antibody which targets an antigen expressed by plasma cells, or removing antibodies by, for instance, immunoabsorption. And what is important is that we got some evidence from these trials that there is maybe a subgroup of patients who would benefit from these therapies. But the trials also showed us that it's not the majority, it's maybe a smaller fraction. And what was also clear is that in many cases, the trials were too small. And this brings me to our concept, the idea here is, so probably only a fraction of the patients has an underlying autoimmune disease. And what's important is that you identify this fraction of patients so that you can stratify treatments and deliver treatments really to those who would benefit from depleting, for instance, antibody-producing plasma cells. But you cannot, and this is what I try to depict here on the right side, rely solely on autoantibody profiles, because what has been shown in addition to G-coupled protein receptor autoantibodies, that there are many others. Classical nucleic acid antibodies, interferon antibodies, thyroid-specific autoantibodies. And this makes it very difficult so at present, we don't have a lead autoantibody, which would allow us to identify maybe the majority of the patients. And this I show here on the bottom. You might have B cells expressing autoantibodies, but targeting different autoantigens. And what might be more important, or in addition, an added value, is to identify the B cells as a whole, which might look different to the ones we see in healthy individuals not suffering from a chronic inflammatory autoimmune process. And how we do it is this depicted here. We always start in my lab with a large-scale immune cell phenotyping, multiplex cytometry. Very often we use mass cytometry. It has a lot of benefits, and I'm happy to answer questions why we do that and not spectral flow. It has a high enough dimensionality so that we get a better picture on the immune cell composition than with classical cytometry, but it is also allowing us to acquire enough cell numbers so that we get a better power on the analysis. And if we know what is altered, we then run cell enrichment for molecular profiling, single-cell RNA sequencing, which will help us to develop hypotheses on underlying molecular processes which go wrong, which will then be tested in 2D and 3D model systems. So that's the principle. And why this is so important to start with a large-scale immune cell phenotyping, explained with an example from accepted classical autoimmune diseases, and that is autoimmune anti-neutrophil cytoplasmic antibody ANCA-associated vasculitis, a disease first targeting the kidney, but it can spread to lungs and other organ systems. There are two main autoantibodies reported, both targeting neutrophil proteins, PR3 and MPO. But the thing is, the patients look clinically distinct, but no one knows why this is the case really, and whether there is a link to an altered B cell activation process. And this was our starting point. And if one enters into the analysis, what might be different in PR3 or MPO driven ANCA, one has to dig into how T cells communicate with the B cells on their way to differentiate into antibody producing plasma cells that could starts at the border of the follicle in the secondary lymphoid organs and involves some co-stimulatory molecules listed here. But some of the B cells becoming activated, they can differentiate at this stage already without further maturation into short-lived plasmablasts, which look completely different to another kind of antibody-producing cell, the long-lived plasma cells, which needs a further maturation step and further engagement with a lot of CD40 ligand interaction between the T cells and the B cells. So you can have in patients different sources of activation of antibody producing cells, and this is what we try to dissect. And what we run there is this multiplex cytometry with mass cytometry with 45 markers, which allows us to really categorize the different B cell and plasma cell population into certain clusters here. And then I can overlay this UMAP, which you will receive at the end, with the origin of the B cells, whether they come from healthy controls in blue and here in the upper UMAP, in the middle, whether they come from MPO positive ANCA patients, or at the bottom from PR3 positive ANCA patients. And what you can see is that the MPO patients really have a completely altered B cell phenotype. And this might also mean that you get other types of plasma cells, other differentiation pathway, which will then allow us to elicit better targeting therapies. And this is exactly what we then did with the after enriching the certain B cell subsets, we run single cell RNA sequencing with also antibody B cell receptor sequencing, and this allowed us to really clarify that the MPO ANCA is more driven by these short-lived plasmablast differentiation, whereas this is different for the PR3 ANCA. This is just an example, and I will now show the results, the first results we have from the ME/CFS patients. And we started this investigation with 4 clear hypotheses: that we believe that in a subset of patients there's an autoimmune process, and that these patients would benefit from autoantibody targeting therapies, and that this subset of patients or subgroup of patients is characterized by distinct T and B cell activation processes, interactions, and that deciphering these processes will help us to develop more better targeting therapies. We did exactly the same as with the ANCA vasculitis patients. We profiled the B cells, and which helped us to define certain meta subgroups of B cells, so whether they are naive or memory B cells, with the markers you see there on the right side, IgM and IgD, highly expressed by naive ones, and the memory B cells lose that, but acquire other markers such as CD27. And if you then again try to see where are the B cells from the controls and the SARS-CoV-2 induced ME/CFS patients located, you can see, to our surprise, clear, distinct accumulations. The B cells of the ME/CFS patients accumulated here at the top in an area quite distinct from the healthy controls, where none of them really accumulated. And this is not just one example. This we observed in 3 study cohorts, so really highly reproducible. What is this difference? If we now zoom into the memory B cells, we can visualize that even more striking, that you have an area which is only occupied by B cells coming from the ME/CFS patients. And when we ask, what we then did is can we use this information and the clusters which are specific for the ME/CFS patients to identify this autoimmune endotype and also elicit more targeted therapies? And for doing that, what I did is I studied patient samples from a small one, but still very helpful immunoabsorption trial where all the antibodies were removed by immunoabsorption from the patients diagnosed with ME/CFS, but also being autoantibody positive. And that this works has been published in a subgroup of patients by Carmen Scheibenbogen last year. What we then asked is whether any of these clusters we detected in this ME/CFS specific region is not only higher in all of the ME/CFS patients, so patients, and this is pre-treatment, either responding or not responding to therapy, or whether there is a specific cluster which is also more enriched in the patients responding to the therapy. And this is indeed the case. This is cluster 11. And this is characterized by a profile which is indicative of activated class-switched memory B cells, and not 100%, but enriched in patients responding to the therapy. So an indicator that there is indeed enhanced activation, memory B cell formation in a subgroup of patients who would benefit from immunoabsorption or plasma cell depletion. And we now try to see whether these patients are also characterized by an activated T cell population, which helps the B cells to become plasma cells, and that's T follicular helper cells. We did something similar with the CD4 positive T cells, and we saw that the cluster which we annotated as TFH cells is higher in a subgroup of patients, and that's exactly, again, the patients responding to a therapy, to immunoabsorption, and the cluster frequency is even reduced after the therapy. So we have in this subgroup of patients a clear indication that there is B cell and T cell activation, and especially B cells and T cells, which are communicating in the process of differentiation of antibody-producing plasma cells. And the last hint comes from profiling of soluble mediators in plasma, in EDTA plasma. And we run ELISA on these samples and asked ourselves whether there's what defines the plasma of ME/CFS patients versus controls, and what is maybe not evident to all of you, but there are many members of the TNF and TNF receptor family, and also CD40 ligand, and these are markers of activated CD4 positive T cells, but there's also BAFF receptor important for B cell activation, again indicating enhanced T B cell communication. And this changes dramatically after immunoabsorption, so the profile is really normalized. Of course, this is not a curative therapy, but it helped us dramatically to understand and to define this autoimmune endotype in the patients. And with this, I summarize. So we think we have more evidence now that there is a subgroup with autoimmune processes and that they would benefit from these therapies. We have first glimpse into alter T and B cell communication, but we are at the moment running the single cell RNA-seq profiling, or they are the data. We have already the data. They are analyzed now, which will then help us to run these mechanistic in vitro studies and hopefully narrow down some new novel targets for these patients. And with this, I'd like to thank you all but of course, my group and there, especially Lukas Arentholz, who has is really focusing a lot and working very hard to get this also into a publication now. Thank you very much.
Thank you, Birgit. That was terrific. Yeah, wonderful overview. We have some questions. So the first is, you mentioned 8% of those infected with COVID develop PCS. Isn't this far too low? Is this figure considering reinfections, or is that a first infection estimate?
Please, I mean, the number might be different. It could be higher. These are figures we collected during the last years, and certainly they don't include all the patients, because this is especially, or until recently, was a big problem in Germany, that many patients are actually not recognized because they don't receive the other or the GPs do not recognize them as PCS patients. So the numbers may be well higher. Yeah, this is one argument. I have to say, I mentioned that to Susan at the beginning. I'm not a clinician. I'm a pure immunologist, but I completely agree. We probably, and we lack real numbers, and this is also important together throughout the world to really get the real numbers.
Thank you. Yes. What is the current statistic for the number of folks with long COVID that also have ME? And can you please elaborate on your statement that most severe form of PCS is ME? I ask because I'm seeing some deliberation within these communities, and it's showing signs of divisive and sometimes harmful rhetoric, which is not reflective of the current consensus under the IACCI umbrella.
Okay, so here I really rely always on the numbers from my collaborating clinicians, and from a big care cohort, which we call PIES Care, initiated last year, at the beginning of last year. We have an estimate that roughly 30% of the PIES patients, and this is not only including SARS-CoV-2 triggered post-acute infection syndromes, develop ME/CFS symptoms. But again, I think here these large prospective cohorts will help us to define the numbers much better.
Thank you. So I'm going to ask the next question in a more hypothetical way. So because we can't really take personal questions, but if someone has very high antibodies to the alpha variant, could this be due to the autoreactivity of B cells?
Yeah, with autoantibodies, it's always the measurement alone and the levels, the quantities are it's not sufficient. What is really important is to dissect and whether they are agonistic or antagonistic, so whether they really interfere with the function of a certain receptor. So that's why, per se, I cannot say that right now. Likely cannot be excluded, but one would need to run further tests.
Great, thank you. I'm going to allow one more question, and then I think the panelists can actually see the questions so they can choose to answer the additional questions that are there. So the last one for you, Birgit, is there any relationship between ME/CFS positive patients' antibody profiles and past history of COVID infections and/or COVID vaccine?
These are really tough epidemiological questions. I have very important, as I said, but I cannot really provide a correct answer. I think that, again, we are lacking data here.
Okay, thank you very much.
But if there is any immune mechanism related question which someone would like to get an answer
That's great. Okay,
um,
so we'll sort out a way for you to make sure you can see these questions, and if you can post some answers, that would be great.
okay.
Thank you very much. Forget that was terrific, and thanks for your responses to those questions. So we're going to turn now to Professor Danny Altmann, who's a professor of immunology and heads a lab at Hammersmith Hospital campus of Imperial College at London in the UK. His expertise is in the HLA system, antigen discovery, T cell subsets, and NK cells. And his title of his talk today is Recent Progress on the Pathophysiology of Long COVID. Thank you very much, Danny, as well, for joining us from overseas. Over to you.
Great pleasure. So, yeah, and so I'll try and follow that super talk, and I might almost convince you that Birgit and I had planned our talks together, because I hope that my talk will be quite complementary to hers and will kind of dovetail quite well. Let me know what you think, whether this works or not. Oh, so. Okay, so hopefully you're seeing that. So that's a picture of Imperial College in West London, and for anybody who wants it, that's my email address and my Blue Sky address. And I
Sorry,
was trying.
Danny. Oh, there you go. It's just switched to full slide. That thanks. Yeah,
Is that okay now?
it's perfect now. Thank you.
Good. Sorry. Yeah. So I'm always available on those addresses and always try and keep in touch with people and communicate with people. And yeah, so like Birgit, I've been on this journey of COVID and long COVID now for many years, and quite early on in spring and summer 2020, when I began to be contacted with people by people, let's go back a bit, who had long COVID and what we came to see as long COVID. I was contacted by a patient and activist, Jez Medinger, and we ended up writing this book together, the Penguin Long COVID Handbook, that encapsulated a lot of the early knowledge. And I put it up here partly because it's a fun book. But if you think it's a kind of big head thing to do to show your book, it's honestly not, because you'll see that this screen grab was done very proudly by me on the first day that it came out on Amazon. And on the first day, I saw you could already buy 2 used copies from people who clearly weren't that impressed by it on the first day. So I hope that some people at least enjoyed it. But do have a read. And it has been a long journey, hasn't it? Obviously, particularly for the sufferers. And in England and the United Kingdom, I guess, as in Canada and Germany and the other countries we're talking about, a sense of some kind of frustration and neglect that the science has moved forward in some ways quite well, and yet the will of the policymakers and the governments to support people with long COVID has just waned in our kind of post-COVID age, where we're told to move on and ignore it and forget about it. So I don't know how familiar and how well kind of English swear words transfer across to Canada, but this was famously were the comments shown at our national COVID inquiry when Boris Johnson, Prime Minister at the time, was being interviewed. And this is the response that he made when he was shown the Department of Health briefing about long COVID, and this was his view of it, which is kind of informative, I think, in terms of the mess we've come to be in all these years later. That I said we do have an immense amount of knowledge about the immunology and pathophysiology of long COVID and ME/CFS, and have lacked the momentum to really address it and do anything sufficient about it. Because this is, again, a slide that I made some years ago, fairly early on, because one of the things that we were quite proud of, I think, in the United Kingdom, that at the time we were kind of ahead of the curve in putting national health government funding into dedicated long COVID clinics up and down the current country. They've kind of waned with time to the point where there are rather few of them now, and many of the patients, I think, barely know whether they're still on their books or not. And all the questions that we hadn't addressed then, we've addressed even less well now. How do you get referred to them? The kind of questions that Birgit was referring to in her talk about the kind of diagnostic and biomarker gaps. Which clinical specialties do we want sitting in them? What kind of specialist equipment or tests do they need? how do you deal with people with a kind of complex multi-system, multi-organ disease, where you might need to be able to move between needing support from rheumatology or cardiology or endocrinology or respiratory teams? And if you could see all of those teams under one roof, which would be the ideal, what type of therapeutics would they offer? And how many years would we need those clinics for? And, I show those slides, not to kind of wind you up or anger you, but just to kind of remind you that those questions have just sat with us all the time, and we need answers to them. And we're kind of, if anything, moving away from the answers, not towards them. I don't need to kind of label this slide. This is my version of the slide that Birgit showed that came from a piece that we wrote in Nature Reviews, where we also tried to pull together the working hypotheses for where long COVID might come from. And by now, these are ideas that will be familiar to all of you. All I'd say is that in the intervening years, evidence has continued to be accrued. I think we sometimes now talk about something like 90,000 different peer-reviewed publications reporting long COVID in the whole of PubMed, which is an awful lot of knowledge, and how poorly have we exploited and translated that knowledge. And I guess the evidence base for these different hypotheses has panned out perhaps slightly variably, some of them more heavily supported than others. And my only regret is that sometimes researchers get siloed into a sort of cult following of one hypothesis or the other, when in fact we're looking at a kind of complex disease, and we need to be looking across these different hypotheses at a range of different mechanisms. And I know I'm sort of preaching to the converted on this, but at the beginning, I used to get approached by lots of patients asking me a very simple question, which is, you're meant to know about post-viral sequelae. From your knowledge of post-viral sequelae, how long will I have this long COVID for? When will it go away? And I was ashamed that at the beginning of the pandemic, I didn't have any good answers for them. So I became very obsessed with trying to track down clinicians who'd looked after people during the SARS-1 outbreak. And obviously that's a story that's very familiar to Canadians because of the University of Toronto outbreak. And this man, John Patguy, was the lead clinician who looked after those patients. So if you've never had him as a guest on your webinar, do invite him, because he's a kind of font of knowledge on this kind of stuff. And I commissioned him to write a review article for this journal at Oxford University Press. And the piece that he wrote was almost sort of so depressing that I hardly dared communicate it to anyone, so sobering, because it finished, I think, with this paragraph in his conclusions, and you can read it for yourself. None of our 50 patients got their life back with time and treatment, never able to return to work. Some had a trial at return to work and failed. He reports a cohort of, I don't want to come scaremonger here, albeit, very sick people who were hospitalized and weren't perhaps typical of an average long COVID cohort. But, 20, 25 years later, these are people who haven't yet got their lives back and are still waiting to resume their baseline health. And a reminder from some of the UK data, but you could tell this story variably from different countries around the world, that, the people who want us to move on and forget about this, I think, imagine that this is a kind of snapshot historical problem from people in 2020 who were perhaps unimmunized, unvaccinated, and exposed to that first variant, the Wuhan HU1 variant, and that therefore the problem will be going away and that the numbers will wane and dwindle. That's not the case at all. So in the UK, one of the things we benefited from was the ONS, Office for National Statistics, data on this, and then subsequently the GP survey data. And what it says about the country that I come from is that we've accrued new long COVID cases through each of the different variant waves, including in the last 3 years or so in the Omicron variant waves. And through that, we've sustained a population of people with long COVID in our country of about 2 million. So that means that some people, and I'll talk about this in a minute, do recover. Not all, but perhaps a minority, but they're replaced by a new wave of long COVID people who, even in multiply vaccinated populations, are still susceptible. So this is a more recent snapshot of that from the GP survey, saying that last year we considered that we were a country where 4.2% of all adults describe themselves as having symptoms of long COVID. That's mind-boggling. I wish I still had the ear of the Prime Minister's office and the Department of Health to rub their noses in that data and ask them if they really want 4.2% of their workforce out of action and out of the economy. So I won't present a lot of immunology data, but we've worked in detail with 3 big cohorts. One is an adult cohort called Wilco, working out the immunology of long COVID. One is a pediatric cohort with the same kind of protocol. And one is Rebracovid, a cohort all the way across Brazil, where they had a slightly different natural history and also a different variant at one stage. So just to, this will be familiar to you, but to tell you a little bit about the cohorts. When you look at working out the immunology of long COVID, the Wilco cohort that we worked with at Hammersmith Hospital, the long COVID people are more likely to be female. They've got a fairly normal BMI, and I do think that to my mind, that sort of female skewing that you're all very well aware of harks back to the kind of stuff that Birgit was talking about, because obviously students of any autoimmune disease know that one of the hallmarks of those autoimmune diseases is that so many of them are female skewed. And we think that's because of the obvious kind of sex hormone imprinting on different aspects of the kind of flavor and program of immune subsets. So we did the same thing in a pediatric cohort, and also were able to do MRI imaging in those, and the same kind of story that they are long COVID patients drawn from each of the variant waves, but big difference because many of them are pre-puberty. There you don't have the female to male bias, and you have kind of equal females and males, or perhaps slightly more males and females, which I think confirms that it's kind of, the obvious kind of sex hormone driven difference. And whether you're talking about adults or children, your chances of having long COVID are to some extent driven by the number of symptoms you had, the severity of symptoms you had, and the length of symptoms you had during your acute infection. So that's not by any means to say that if you had a mild infection or even an asymptomatic infection, you can't have long COVID, but your risk of having long COVID was certainly increased by having a sort of more durable, more severe acute infection. And I show this slide not because you won't all be familiar with the kind of range of symptoms in long COVID, but just to make the point that I think that for a while we talked a lot about the enormous symptom list, didn't we, of this condition where you can have 200 different symptoms described. And sometimes I think that confuses people we're talking to, who think it must be something terribly complex and vague and poorly described and heterogeneous. And I feel like the more people that we see and that different cohorts describe in the world, sure, there are many symptoms out there, but I'd argue that, the big symptoms are big, the post-exertional malaise, the fatigue, the wheezing, the breathlessness, the neurocognitive symptoms are kind of common and head and shoulders above the others in terms of helping us to describe this condition. And again, like I said, it's similar in our pediatric cohort as in our adult cohort. And I simply show this because to kind of air my grief to you that when we were doing this study in the UK, we became very close to having our funding withdrawn by the government for the pediatric cohort, because even after the grant had been awarded and funded, because they said, we've been told by our advisors that you won't have any children to study because children don't get long COVID, they recover spontaneously. It's just a bit of exhaustion. And, we have very, very sick children many, many years out. We also do a lot of immunology on the immune subsets and RNA-seq and immune responses. And this is a very complicated slide of all of the spots are measuring T cell responsiveness to different antigens. And to cut a long story short, if you compare immune responses, T cell immune responses, either to different parts of SARS-CoV-2 or to other antigens, that little thing in the middle there that says CEF stands for CMV, Epstein-Barr virus, and influenza. So it's measuring immune responses to other viruses. We don't see a very strong signal most of the time that children with long COVID have either altered immunity to their COVID or to other viruses. We don't, I know there's a view out there that's slightly popular that COVID-19 is a kind of immunosuppressive virus that shoots your immune system to pieces and makes you very, very vulnerable to other viruses. We simply don't see that signal in our cohorts and are not particularly sort of keen aficionados of that view. I did want to say, though, that in our cohorts, adult and children, we survey them longitudinally over many years now, and we present a picture I think will be familiar to many people on this webinar, that you can think of a long COVID cohort as perhaps 40 or 50% who are recovering or have recovered with time. So you're not stuck with it necessarily forever if you're one of the lucky ones. And I can think of many people in our study who have returned to baseline health and are now riding their bicycles and riding their motorbikes and returned to full-time work. And I can think of about another third or so who have essentially stayed the same with time and are still very much having problems and haven't returned to baseline health. And then another third or so who have worsened with time and are in real trouble and don't feel like they're getting better and don't feel like they're getting their life back. And we've done a ton of demographic analysis and epidemiology and immunology and molecular immunology to try and work out the correlates of what predicts you being a kind of got better versus got worse person. And I wish I could tell you the answer, but I haven't got a simple answer yet. But I really hope that we will have. But just to say that, one of the biggest markers that you all understand is that, we really can't trivialize long COVID, because one of the biggest differences is that if you've got long COVID, you are really likely to have either lost your job, faced early retirement, been made redundant, or been forced into a part-time role. And that is such a big statistic. It's huge. Like Birgit, we're very keen on the idea that one of the key things we need to understand in the mechanism of this disease is that in some ways it looks like other autoimmune diseases. And I'm a person who spent quite a lot of my life studying things like multiple sclerosis and type 1 diabetes and rheumatoid arthritis, classic textbook autoimmune diseases. And the reason that I make that link is two reasons. One is from population data, sort of national data, that says that if you had SARS-CoV-2 during that early wave, you have an increased risk of a classic autoimmune diagnosis subsequently. And then we can do the kind of things you were hearing about just now, where we use panels of self-antigens to screen for autoantibodies and show that long COVID people look different to healthy controls who made a full recovery. So this is from that piece we wrote in Nature Reviews in Immunology. If you fancy that kind of stuff, do go away and have a look at it, saying that lots of people have looked at lots of different autoantibody panels and find them raised in long COVID. So that's a kind of, if you like, a kind of conventional hospital medicine way of looking. This slide I find very impressive. It's not from me. It's a few other teams have looked at this. And what this says is that if you look at the very clean data sets from early on, if you think about in 2020 or 2020 to 2021, the world of COVID analysis was still, if you like, a simple world where most people didn't have vaccines on board yet. And most people who had COVID were accessing either a PCR test or a lateral flow test. So we could divide planet Earth up in electronic healthcare records into people who we knew had COVID and people we knew hadn't had COVID at that stage, okay? And if you follow the national healthcare records in subsequent years and say, what happened to those two pots of people? What was their risk of disease subsequently? And the answer is really striking, that they have an increased risk after COVID of a number of different diseases, But if you have a look at the risk of classic autoimmune diseases, things like type 1 diabetes or lupus or vasculitis, there's a 2 to 3-fold risk of a subsequent diagnosis. Okay. So having COVID-19 on board has done something to your immune program and regulation of your immune program that's driven you into an autoimmune disease. So scientists and immunologists like to use a lot of fancy technologies, and we call them omics. And I think the sort of new kid on the block is looking at that in terms of autoantibody analysis. So we've looked at it, for at autoantibodies one way or another, probably for the past 60 or 70 years. But if you wanted to look at it comprehensively, you'd think, well, actually, the human body comprises, we can never really agree on the number, but call it 25 or 30,000 different proteins. Potentially, we could be autoreactive, autoimmune to any of those. How are we going to look at that comprehensively? And labs have developed a lot of technologies for looking at that. And we use a sort of whole proteome array to look at autoantibodies to everything. And that's a little sort of cartoon of how we do it. And to cut a long story short, the simple answer is that people with long COVID have clusters of autoantibodies that are big clusters and diverse clusters to lots of different parts of the body. Healthy people who don't have long COVID also have some of these, but far fewer. And we're desperate to find the therapeutics that'll help to turn off those autoimmune responses, because we're also confident this is one of the routes we can take to switching off long COVID. And I guess I'd make the claim that immunologists have a track record in this area. Yeah. So through my career, things that used to be thought really poorly treatable, like multiple sclerosis and rheumatoid arthritis and type 1 diabetes, have had a load of different biologics, different antibodies come on board that can treat them rather more effectively than we used to do, and make people's lives quite a lot more livable. So I feel very kind of gung-ho that we can do well on this. I just want to finish by a few kind of summary points from this conference. So I was the co-organizer together with Akiko Iwazaki and Avi Nath in the summer of this Keystone Symposium, where we got together lots of big thinkers in the world in the field of post-viral sequelae, long COVID and other post-acute infectious syndromes. And I just want to finish by giving you the flavor of where we are. So if I was going to give you a sort of quick summary of it, I'd say that, we've talked how we need biomarkers. We need biomarkers for clinical trials. We need biomarkers for general doctors to refer people into care pathways. And I think that our inability to knock people's heads together and agree the right biomarkers for diagnostics and study recruitment and things has been a real problem. And I think we're trying at the moment to get people together to do better. I do think the field is coming of age, and we have a lot of people doing this kind of stuff now. We have a lot of good cohorts. Another source of frustration to me halfway down the slide is that, we all concede that we have this very heterogeneous condition. And, for heaven's sake, we're in the age of machine learning and artificial intelligence. We should be ashamed of ourselves that we haven't done better in terms of making sense of these patterns and getting people into the right clusters that have, biological and clinical meaning. We have very diverse approaches to clinics around the world, most of them suboptimal, but we also have some very clever clinicians in this field who are doing some very fancy stuff, and we can really learn from them, because those are kind of human experiments in action where you can learn from what works best. You've heard a lot, I know, about virus persistence as one of the hypotheses on the table. It's appealing because it would offer a simple explanation for why 400 million people on the planet still have long COVID. And we ought to be able to make them better with antivirals. But again, you probably know that there have been 4 trials so far using Paxlovid as an antiviral, and the results have been a little bit underwhelming. And again, we need to look at that data very carefully and work out how to do better. There's a lot of words on that slide, so but we talk about things like data on latent herpes virus reactivation, autoimmunity you've heard about, immune perturbation, a lot of progress, I think, from animal models, a lot of progress from comparing with other post-viral conditions, things like chikungunya and glandular fever, and even Lyme disease. And new technologies coming on board. For you, people who like this kind of thing, do go and have a look at this paper from Mike Peluso's lab in UCSF that a lot of people like, because those scary pictures on the right are a new approach that they use at UCSF for PET imaging to show immune activation in the body, and as a sort of marker of long COVID. We're certainly in a phase at the moment where we're trying to pull together cohorts from long COVID and ME to try and look at commonalities between those two conditions. So there's a lot of detail on the slide, but the study that we started recently is a sort of next generation study that we call Rosetta Stone, where the analogy will be obvious, just that we know a lot of things about ME/CFS. We know similar and different things about long COVID. And if we could actually pull together that knowledge from cohorts of both of them analyzed side by side, we think we could do our decoding much better. So at the moment, we're collecting blood cells for multiparameter flow cytometry and RNA-seq and metabolic studies, and stool samples for microbiota studies, and tissue samples for persistent virus studies, and trying to put it all together to just tell the story better and actually try and do better at making some people better. So that's a sort of very speedy tour through everything we're trying to do, and those are the people who've done it. The person, lots of good people here, but the person on the right in the stripy shirt is Rosemary Boyton, with whom I run the lab and run all these studies together. And very happy to take questions.
Thank you. That was excellent. Much appreciated. A number of questions, and I just wonder if Brigitte and Danny, you might be willing to stay for a few more minutes after 2 p.m. our time. I promise we won't go more than 3 or 4 minutes over. So, okay, we'll get started. In Canada, testing is no longer widely available. I'm wondering if this is the same in the UK. Are there concerns about how the lack of available testing will impact the ability to identify new long COVID patients, and how this will impact progress of research?
Yeah, well, should I go first, Birgit? Yes, enormously, yes. So the situation in the UK, obviously, is that, like many other countries, the government and National Health Service haven't supplied free tests for some years now, and they're available quite cheaply at pharmacies. And the proportion of people I know who test themselves when they have COVID symptoms is probably 10 or 15%. So it is absolutely certain that there are huge numbers of underreported COVID cases all around us, and every time you get on a train or a bus. And we know from data from St. Louis from the VRC that your risk today of getting long COVID after a COVID infection, even as a previously infected person, even as a vaccinated person, is probably about 10% of all infectious events. So we're still accruing new long COVID cases, even though we don't always know it.
Birgit, would you like to comment on the status in Germany?
Actually, I don't think you get the tests reimbursed here. So basically, you have to pay it yourself. And from what I get here around, there aren't many people testing themselves. So we are in an environment as before with other respiratory viruses where you don't know who has what sitting next to you in the bus.
Thank you. Next question. When we discuss patients self-reporting recovery, is this true recovery or potentially remission? I believe we have seen from the history of ME/CFS that some patients perceive recovery only to relapse later. Do we know enough yet to make the distinction?
Yeah, I think that's a really good and important question. So the first thing to say is that we shouldn't denigrate ourselves or our community with the words self-reported, as if that makes it less real. Yeah, because self-reported is kind of all we have. because most of the 400 million people in the world can't get to see a specialist clinician who can report it for them or would have the test to report it for them, even if we ask them. But if you sort of put that to one side, I completely agree with the premise of the question. So in the UK, we worked a lot with one of the sort of best cohorts, which was called FOSP-COVID, which was a sort of national initiative to collect all of the year one hospitalized COVID patients and track them for the first 2 years and see what became of them. And that generated a lot of data and a lot of publications, including long COVID publications. And we then tried to go into the data set and do the bee that we had in our bonnet of analyzing the recovered people compared to the not recovered people and seeing what was different about them immunologically. And sure enough, we found that we had to lose a lot of data because people who'd reported themselves as being recovered, when we looked objectively at their measurements, weren't really recovered at all. They were adapted rather than adapted to their disability rather than recovered. So it's a thing that we have to keep an eye on because people are very resilient and they'll try and get on with their lives, but it doesn't mean they're completely better.
And I would like to add, because this is, I completely agree, it's an absolutely important question, and that patients go into remission and can relapse again. We see from other diseases. This is nothing special to post-acute infection syndromes, and one should really bear in mind. And especially the policymakers, because it may well be that in half a year the patient becomes symptomatic again. This is nothing special. This is typical for many diseases.
Thank you. The question is, this may be a loaded question. I think it's a large question, so maybe you could see how best to answer it efficiently here at the time. But what triggers the random inflammation on long COVID, which can cause an array of symptoms on top of the organ and system damage, and how can we address it?
Oh, so I'll start while Birgit's thinking. But that kind of goes to the heart of every paper we ever write and every grant application we ever write. That's like, tell me the whole story. And we talk about things like the ability of having SARS-CoV-2 on board to disrupt immune subsets and disrupt regulation, and the kind of data that Birgit was showing so beautifully of stimulating and expanding particular B cell subsets at the expense of others, and the ability to drive inflammatory cytokines, and the ability to reactivate endogenous herpes viruses, things like Epstein-Barr virus, which in itself goes on to do loads of stuff, including activating B cells and activating autoimmune B cells. So, how big is your textbook of immunology? You could go on and on.
Yeah, absolutely. I mean, the problem with SARS-CoV-2, as I see it, is that you can get replication at many sites, which drives a systemic response. And this is why also when you then develop dysregulations, you can get dysregulations involving different organ systems, and therefore you develop this multitude on symptoms. It is something. I think there is something special of SARS-CoV-2, where other infections are more confined to some local environments. But here, due to this distribution, you drive systemic responses.
Thank you. Last question, and also not a small one, but given your presentations and all of your work on immunology, are there any particular interventions or treatments for which you might be excited or hopeful?
Well, I'll kick off, and I think it's just like the obvious conclusion from both of the talks you heard this evening. That I think that immunologists are quite an optimistic, exuberant bunch, and we've got good reason to be optimistic because we've been one of the great success stories of molecular medicine of the last 50 years, and we've made a lot of people better. If you think about cancer immunotherapy or autoimmune disease and the outreach of immunologics. So I think, we're good at doing this stuff, and we're very close, I think, to a tipping point where we understand enough about the mechanism to begin to make a difference. And, the kind of places I'd look for excitement would be in manipulating autoimmune B cells or manipulating, reservoirs of persistent virus. And I think one way or another we'll win.
Yeah, I think we both agree a lot. The thing is, and we have now a collection of many different fantastic therapies developed for other diseases, which, if we understand better the individual patient case, can reallocate. And so I wouldn't name a particular one because there are really many ones, even for the two processes I described. There are specific ones already developed. We just need to stratify the patients, and that will be the important task for us in the next years.
Thank you. I'll just, we'll close here, but I just want to say a very huge thank you to both of you, the speakers, for joining us today from across the sea. And terrific that you're continuing on with this really important work for all of us. So thank you so much. And thanks to everyone for participating. Please do fill in our little evaluation to help us plan future webinars. So all the best and enjoy the rest of winter, early spring, wherever you are. Take care. Thank you.
Thank you.